Welcome back. We are here with Elinor Karlsson, and we're going to just touch base on some of the interesting things you brought up in the first part of your talk. There was a lot of interesting conversation on Twitter, and one of them was you pointed out that, hey, dogs were third when it came to the genome, right? Humans, then mice, then or a mouse, and then a dog.
Yeah.
And so why was that? Is it possibly because humans and dogs share an environment? Why was the dog third?
That's actually a really interesting question. And I think it actually points to the fact that dogs do seem to have kind of a unique relationship with people. It's something that people really like. One of the nice things about working in dogs as a science is that it's a little bit more accessible to people. But I think there were a lot of very compelling reasons to do it for medical research as well. One of the things I pointed out at the beginning of my talk was that we share a lot of diseases with dogs, and that includes things like cancer and diabetes and things that are huge problems for people as well. And in dogs, we have this kind of unusual situation where we have all of these purebred dog populations that can be very useful for studying diseases like cancer. And so I think the idea was that by sequencing the dog genome, we would actually enable genetic studies of diseases within dog breeds.
Yep, that makes sense. You were also, another question from Melissa on Twitter, noted that you were very careful to say that nearly every cell has copies of the genome. So which cells don't have the genome, and what is the deal with those cells?
Oh, you guys are going to just totally push my remembering of basic biology. Which of the blood cells is it?
Yes.
Which ones is it? The platelet? Red blood cells.
Red blood cells.
Yes. So it turns out that in
crowdsourcing answers.
It was a long time ago that I took biology, okay? This is why I have other people to turn to. So red blood cells do not have a nucleus, and the nucleus is the part of the cell that carries the DNA. Okay. This turns out to be true, I think, of all mammals, but it is not true of all animals. And so it turns out that when you're working in other species, I think I'm trying to remember if this was true of the rattlesnakes, because we have a project going on in rattlesnakes.
Birds and reptiles get a bit weird with
reptiles, crocodiles, yeah. Anyway, so they do have a nucleus in their red blood cells, so they do carry a copy of their DNA, and it actually means you get way more DNA out of their samples than out of mammalian samples. They're much nicer. Yeah.
More DNA. All right. Well, those were kind of the basic genetic questions that were coming through.
So no more basic biology.
We are done.
Okay, good.
I am done with that. Yeah.
Now, like the topic that really took off on Twitter during your talk was the whole link between ear pigment and hearing.
Oh, yeah.
So I've got a multi-chain kind of question scenario going on here. Yeah. Starting off with, I guess, can you elaborate in relation to the hearing and ear pigment link? Can you elaborate why the pigment is necessary? And is this just for boxers? Is it for all dogs? Is it for all mammals?
So the reason, if I remember correctly from when we were working on this project, is that it doesn't actually have anything to do with pigmentation. It turns out, as with many things in the genome and in our body, pigment cells have multiple, not just the pigment cells, but they actually have multiple different jobs. And so within the ear, it doesn't matter what color inside your ear is, but it actually acts as a kind of an ion channel for moving things across a membrane. And so if you don't have it, that doesn't happen, and I think you can't hear. But it's not true of even all white dogs, let alone all mammals. And so it turns out that the reason why these dogs are deaf is because they don't have the pigment cells in their ears. But there are actually multiple different ways to have white fur. And so these dogs have white fur because they don't have any pigment cells. You can have pigment cells but not produce pigment, in which case you're not going to have any problems with your hearing, but you'll be white because you can't actually make the pigment from your pigment cells.
So that will probably help out Kim and also Dale, because they asked, and I might offer both at the same time, then let you respond. Would the colour gene be different in a breed like Bichon Friesers, where they're all white without the tendency to be deaf? And Dale mentioned if breeders are selecting for lighter colours, are they then on their way to creating deaf dogs?
Yeah, so the answer to that second question is definitely not.
Yep.
And we actually ran into this because we have this citizen science project, Darwin's Dogs, where we ask people questions about their dogs. And one of the questions we asked was, is your dog, how much white fur does your dog have? And we had the whole set of 6 pictures, and you had to pick out which one your dog was. And then we did the genetic association study and looked at which genes were connected to being white, and we didn't find mid-F. And of course, this being the first project I worked on, I was like, oh God, I've got it wrong. It's something terribly. But then my graduate student went back and looked at the photos that people had given us of their dogs. And it turns out that many of the dogs that people were saying were white they were white. They were just cream, which is a different kind of white. So if you're
yeah.
cream, you actually are making pigment. You're just making light pigment.
Julie's looking at painting her apartment, and
Yeah,
white has
see?
been all the shades has been a very hot topic. Yeah,
And I mean, I love that situation because to me it says that we have to be really careful about the questions we ask. Like they were answering completely accurately. They had a white dog. It's just wasn't the white we were asking about.
It was more an antique white.
Right.
Yeah, exactly. We should send them little pink swabs and have
Quarter
them
hugs
hold
bristle.
on to
Yeah. Okay, so then I guess you said that 2 out of 100 white boxer dogs are deaf. So my next query is then, if all 100 dogs have the same genotype, why aren't all 100 dogs deaf?
I think that's a fantastic question, and the answer
It's
is
actually not mine.
whoever answered it, asked it, great question. The short answer is we don't know.
Yeah.
We actually had a lot of discussions around this. It's possible there are other changes in the genome somewhere else that actually also influence the migration of the pigment cells that affects that. But another possibility, and this is something that sometimes people don't think about, is that there's just what we call noise. And so we've broken something, we've changed something so that the gene doesn't get turned all the way up, but maybe with that, in the absence of anything else, so just a very low level of the gene gets made into protein. That's just kind of the default state, and it just varies a little bit from dog to dog, just by chance. I don't know the answer to that, and I'd actually be really interested to see whether we can figure that out. Maybe if we had enough dogs, we could compare the white dogs that were deaf to the white dogs that weren't deaf, and actually see if we could see differences genetically between them.
Do you think it's a type of phenomena that could apply to other phenotypes as well?
Definitely. So I think a lot of times when we think about genetics, we think of it as being an on or an off. If you have this, then you will have this. If you have that, then you'll have that. And it almost never works that way.
Not that simple.
And one of the things we found out really early in dog genetics is even when people came to us and they said, we have this really simple trait, it definitely behaves like this. And then we would start studying it and we were like, it turns out much more complicated than that. So I think it's definitely much more the rule than the exception.
So it seems like you are pretty excited about whole genome sequencing.
Yes, I as a computational biologist, I love data.
Yes.
Can you explain to us, I guess, what it offers?
Yeah.
Like why be excited?
Yeah, so, you know, in the past, we didn't do whole genome sequencing because it was very expensive. It generates a huge amount of data, and it turns out that then you have to put it somewhere, and you have to pay to put it somewhere, and then you need really, really big computers to analyze it, and then you have to have conversations with Google and all this stuff. So there's a lot of complications in having that much data, but it also gives you all the information that's in there. And so one of the problems we've had with using what we call these SNP arrays, which are testing maybe 100 or 200,000 positions across the genome, so only 100 or 200,000 out of the 2.4 billion that are there, is that the way we picked those SNPs was by comparing, for the most part, by comparing purebred dogs to one another and looking for differences between them. And that means that sometimes those SNP chips are not as useful for studying dogs that don't have anything to do with purebred dogs. So say village dogs in Asia that aren't related, you know, that aren't part of that purebred lineage, you may not actually be looking at differences that are important in those dogs. And so by doing whole genome sequencing, you actually get all of the information. And so you don't have to worry about having those kind of biases in your data set.
Well, speaking of biases, as somebody that came to this not with necessarily a big thought about dogs, what has surprised you the most?
I think recently the thing that I have started thinking about the most, and it's partly because we've been doing this project where we've asked people to try and guess what breeds are in purebred dogs, in mutts, which got me thinking a little bit more about it, is the way that everybody walks up to every dog and the first question they ask is, what breed is it?
What is that?
Oh,
and I find that just fascinating.
I have a big Muppety Wombly crossbreed, and that is, I seriously, I need a T-shirt because all the time we take him out in public and it's, what is he?
Yeah.
Because he's just big and fluffy
And then you start to wonder, like, once people have decided what it is, how does that then change the way they relate to the dog? Do they have a different relationship with the dog if they think it's X versus Y based on their ideas about what this breed is versus that breed?
Right.
And how much does that actually affect what we're actually seeing when we look at that dog? Which is a whole very complicated question. But I think it gets back to that fact that I'm a geneticist. I'm really interested in understanding how DNA affects complicated things like behavior. But that's never going to be the whole story. And we don't even know how much of the story it is because environment is such a huge part of personality and behavior and who a dog actually ends up being as an individual. And so with the genetics, it's an important part of the story, but it's really only the first part, and looking both at the environmental part and also how the two interact. I mean, maybe if you're born with a certain genetic personality, then stressful events are going to affect you differently than if you're born with a different kind of personality. And then those two things are so intertwined with one another.
Right. So just in terms of your steps into the world of behavior and ethology and how one collects or observes behavior, has anything surprised you about that realm of looking at dogs?
To be totally frank here, the first part of my step into this world was when somebody in my group, Catherine Lord, had to explain to me what ethology was.
Certainly.
That was an important first
Welcome
step.
to the field. Yeah.
Then I had to learn out about coding behavior.
Right.
And then Catherine has been working on this project studying wolf puppy behavior to me, where she administers this how the puppy reacts to novelty at different time points during development, during early puppyhood. And the thing is that you can only do the test on each puppy once, because once you've exposed them to the novel object, they've then been exposed, so it's not novel anymore. And which means that if you have a litter of like 6 puppies and you want to capture weekly time points, 1 time point per week from when they're like 2 weeks old to 8 weeks old, you have 1 puppy 1 week, and you cannot screw up that test. I'm coming from a field where we've got 2.4 billion letters in the genome, and we sequence thousands of dogs, hopefully one day, at least hundreds now. And so we've got so much data. The idea that everything's that you have to be so careful about what you do because you only have that one opportunity has been a really kind of interesting change in mindset for me.
Yeah. Well, we also will post those great videos of that you guys have of the wolf pups.
That was, exactly. So one of the things I realized was when I was finishing up my postdoc, which is what you do after you do your PhD, before I started my research lab, was that I was a computational biologist, which meant that we got samples and somebody in the lab dealt with them, and they got sent off and they got sequenced, and then I got data on my computer that I then analyzed on my computer, and then I would email it to people or write things about it on my computer. Every once in a while I would print something out onto a piece of paper and I'd have something to hold. But other than that, my entire world was virtual. Like nothing I was doing that involved touching anything. And so as I started up my own research group, it's been amazing to kind of bring people in that means I get to go visit wolf puppies. It's just like the coolest thing.
Julie has said for a long time that canine science is like a gateway drug to science for people, because you pull them in with the dogs and the wolves, and then you hook them in with all the other science on the side.
And then we introduce them to the really, really awful stuff a little bit later. We're like, and by the way, you now have to learn this really weird program.
And some complicated statistics, but whatever.
But puppies, really.
It's all about puppies.
Now, just in our last little couple of minutes, you've got an exciting project that finishes off sort of later on this month that people can participate in.
Yes. So I'll talk about it a little bit at the end of my talk, but we've been sequencing mutts. So one of the things that we started doing actually just after I started my research lab was I realized that I wanted to study all dogs. Partly this was because we'd studied purebred dogs for many years, and I'd have these moments where people would come up to me and tell me that their dog had maybe cancer or they had a really interesting trait, and we'd have to say, what breed is your dog? And if it was the wrong breed or was a mutt, we'd say, no, they're not useful for our study. It turns out that a much more fun way to approach this is that everybody that comes up to you, you tell them that their dog is actually special and wonderful, and you really, really want to have them involved in your research. It's a way more fun to do science that way. But as I kind of got into this, and it's also a way to get much bigger sample sizes very quickly, of course, I realized that nobody had ever actually done whole genome sequencing, as far as I knew, on a mixed breed dog, just a mutt. And so we took a bunch of dogs. We started with 21, and it's grown since then, and the only requirement was you had to have a dog that you had no idea what it was, and you had to run into me within this 2-week period when I was looking for dogs. And it may have just happened to coincidentally include my sister's dogs, who, by the way, I think may now be the most sequenced dogs on the planet, because every time we need to validate a new technology, I go over to her house and swab her dogs. I'm like, we need something to validate this.
My mum's dog is like that in our research group. Back in Australia, we called it the Haiti effect. Like she's participated in every study.
It's like we need a control. So we've been doing all that. So we actually took a bunch of dogs. We developed a whole algorithm for figuring out what breeds were in these mutts. They're way more mixed up than I was actually expecting them to be. But we have the answers now from the genetics. And we thought, well, wouldn't this be a really fun opportunity to find out how well people can actually do this thing they do every time they meet a mutt, which is ask what breed is in that dog and then make guesses. So about a month and a half ago, we launched this project called Mutt Mix. and the website is muttmix.org. And it's just 31 mutts, and we ask everybody to guess what they think the top 3 breeds are in that mutt. And when the surveys close in the middle of this month, we'll send people an email where they can actually go and get their results and see how well they did compared to what we got out of the genetics. And I think it's going to be a lot of fun. It's proven to be very popular. We've got, I think, I just looked, and it's about 30,000 people have participated at this point, which is fantastic.
That's a few. That's
Yeah.
a few.
It's been kind of overwhelming. We had to upgrade our video server a couple times because it turned out it was a lot more people than we were expecting, which is going to mean that we can ask all sorts of questions we couldn't have asked if we'd only had a small number of people. Like, if a dog has floppy ears, do people guess particular breeds and things like that? It could be a really kind of interesting thing to start looking into. But it's been interesting because people kind of keep asking me, why are we doing this? And it was like, well, I'm just curious. It just seems like such an interesting opportunity to find out what's going on here. So
we are very excited you're doing this, and we will pull this more into the panel discussion as well. But we will be back in 5 minutes for the second part of your talk. And thank you so much for this Q&A.
thank you.
Thanks. We'll see you in 5 minutes.